makes these proteins. And so first of all, this is what we call the universal genetic code. All life, it doesn't matter whether you're a bacteria or a human or a tree, AUG codes for methionine. Doesn't matter what life form you are, it all uses the same language. Okay? This next one, where it says, describe the role of ribosomes, ER, Golgi, and the nucleus in the production of specific types of proteins. This is actually a review. We already talked about this, didn't we? Back when we talked about parts of the cells. We already know this, but let's just review it anyway. Remember, the nucleus holds the DNA that codes for protein, and maybe we can draw a line pointing to the nucleus, okay? Transcription happens where DNA is converted into mRNA. The mRNA then travels from the nucleus to the ribosome. Let's draw an arrow to here. The ribosome is where the protein is made. Let's draw an arrow from the ribosome down to here. Do you remember that image? tRNA has an anticodon that base pairs with the codon. Okay, so if the codon is AUG, then the anticodon is AC, right? AUG with UAC. Maybe we should write that. Let's write that. AUG. There's our codon. And then UAC would be the anticodon, right? A sticks to U sticks to A, G sticks to C. Okay, and that brings the correct amino acid. The ER is where the protein travels through and leaves in a vesicle. Remember we said the ER is kind of like a highway for proteins. They can get quickly throughout the body of the cell. And then lastly, the Golgi apparatus is a place where the proteins get labeled. Remember we said it's kind of like the post office. We label things, and because of the labels and the zip codes, they end up going to the correct destination. This here below is a blow up of what's shown above. Let's remind ourselves what all these parts are. This is the ribosome. We said it looks like a hamburger bun, right? This molecule here, the long one, remember that is the messenger RNA. That's a copy of one of the genes from the DNA. Remember when we have 1, 2, 3 nucleotides, we call that a codon. Think of it as like a little DNA word, right? It's like a word. Up here we have tRNA. Kind of looks like the letter T, doesn't it? And on the end of the tRNA is the anticodon. It's what sticks to the codon. It's the opposite. Anti means opposite, right? If you're anti-school, you're opposite school. If you're anti-Kramer, you're opposite Kramer. Whatever I think, you think the opposite, right? Okay, continuing on here, here we have our amino acid that the tRNA brought. Here we see a bond forming between that amino acid and this guy. This is the, what's a polymer amino acids called? You know that. You got it? A protein. Okay, so lots of things to label here. That process is what we call translation. Translation. We're going from a nucleic acid sequence to an amino acid sequence. Remember, those proteins are tools, right? They're going to fold into hammers and chainsaws and screwdrivers, and they're going to do the work of the cell. So let's go down below and transcribe and translate the following gene into the corresponding amino acid sequence. Remember how this works? So the first thing we have to do is put the complementary mRNA down. Okay, and this can be a little confusing. So I like to do it one at a time. So the first one is U, and then I cross it out. The next one is U, then I cross it out. The next one is U, then I cross it out. Then the next one is a G, and I cross it out. And the next one's an A, and I cross it out. Then a U, and I cross it out. And a G, I cross it out. Then a C, cross it out. Oh boy. U, thanks, Mr. Scope. An A, a C, C, A,
Okay, thanks. Bye.
G, A, U, 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 G, A, oh man, and then 4 U's at the end, U, 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 U. That would be the mRNA, right? So we put the complementary nucleotides down. Remember, there's no such thing as thymine. When we write messenger RNA, that gets replaced by uracil, right? So it's not apples in the tree, it's Army uniform, right? A with U. Then we have to look for the first codon, which is August, right? AUG. So let's box in or frame the first codon, AUG. And so that tells us where the first codon is, and then every 3 afterwards is going to be a codon. Remember, codons are always 3 nucleotides long. And this one is going to bridge the gap here. Okay. And so is this one. Okay, and if there's some leftovers, no big deal, right? Remember doing this? Okay, good. And so you'll have the chart. You'll get the chart on the Keystone exam. Okay, let's go ahead and use it. AUG is the first one. So remember, left side is the first base. Top side is the second base. And then that tells us that it's somewhere in here. And then the third base is G. And so AUG is methionine, which we can abbreviate MET. Does anybody see CUC? What would be the amino acid for CUC? Okay, C's on the left side, U's on the top side, so that means it's somewhere here. C's on the right side, there it is. CUC codes for leucine, and you could write out LEUCINE if you want to, or you could just abbreviate it LEU.
We have about 1 minute.
Okay, we'll just end right here, guys. We're not allowed to do any homework, so